DNA replication is the copying of double-stranded DNA. It is necessary for cell reproduction prior to cell division. The first step of DNA replication is when enzymes called helicases separate DNA strands. Helicases move along DNA molecules, breaking hydrogen bonds between complementary nitrogenous bases. This allows two DNA molecules of double helix to separate from each other. This creates Y-shaped regions. The second step of DNA replication begins when DNA polymerases add complementary nucleotides to each original strand. Covalent bonds form between adjacent nucleotides. Covalent bonds also form between deoxyribose sugar of one nucleotide and phosphate group of the next strand. Hydrogen bonds form between complementary nitrogenous bases on original and new DNA strands. The third step of DNA replication is when DNA polymers finish replicating DNA and fall off. Two separate and identical DNA move to new cells. Mutations can occur in DNA when DNA polymerase makes a mistake and pair things wrong. Mutations can lead to many forms of cancer. It can affect cell division, which can cause a tumor to grow. When DNA is damaged, it fixes itself. The repair mechanism recognizes the damage. Then, it removes the incorrect or damaged nucleotides. The undamaged strand serves as a template for replacement for appropriate intact nucleotides. Deoxyribose is a 5-carbon sugar and is a component of DNA nucleotides. Nucleotides are molecules that make up structural units of DNA and RNA. It is composed of one phosphate, one deoxyribose molecule, and one nitrogenous base. A replication fork is a junction where the double-stranded DNA splits apart into two separate strands. The helicase opens up to DNA duplex. This creates two replication forks. Then polymerases start to build new daughter chains, and the forks get further and further away from each other. They can make two single strands of DNA. Enzymes are a type of protein or RNA molecule that speeds up metabolic reactions in plant and animals without being permanently changed or destroyed. This is a single DNA strand. The outer layer is made up of phosphate, while the inside is like a ladder. A guanine base pair can only pair with a cytosine base pair, and an adenine base pair can only attach to a thymine base pair. In this step, the helicase is separating the DNA strand. In the next step, the DNA molecule is beginning to separate, preparing itself for a new DNA strand. As you can see, the DNA polymerases are adding complementary nucleotides to each original strand of DNA. This creates a new DNA molecule coiled around the original. Finally, two identical DNA molecules were produced.